Welcome to part 2 of chapter 3 corresponding to the course Human Anatomy and Physiology based on the Marieb cell book. This chapter is related to the cells, and in part 2 we are going to look more closely at the resting membrane potential of the cell. We will review cellular structures and we'll be talking about the life of the cell, particularly the cell cycle, phases of the cell cycle, cell division, and briefly review genetic processes such as DNA replication, transcription, and translation. Okay, so in part 1 we talked quite a lot about the membrane, the plasma membrane. We talked about the different transport mechanisms across the membrane. We talked about the movement of water across the membrane and how it could result in changes in the volume of the cell. We mentioned that there were passive and active transport, and in the particular case of charged particles such as ions, there was a need for transporter channels and transporters, so carriers. And we talked about different types of channels. We talked about some channels being always open, called the leakage channels, and the so-called gated channels that would open or close depending on different stimuli. And we also mentioned that in the case of moving molecules against the concentration gradient, energy was required, so that was an active process, and exemplified one of these active transporter proteins as the sodium-potassium ATPase, or sodium-potassium pump. And this particular transporter is going to be very important to the next topic, which is the topic of the membrane potential. So there are instruments that you are actually able to introduce an electrode inside the cell and have another outside the cell and measure the difference of charge between the inside and the outside of the cell, and you will see that even in the resting state, all cells are going to have a membrane potential, and this is called the resting membrane potential. So this is a voltage that is measured in all the cells, and it has a negative value, which means that the cell tends to be negative. And usually the most common number is minus 90 millivolts. Certain cells can go minus 7 all the way from minus 50 to minus 100. And it's going to depend what kind of cells and what do they do. Be aware that because this is a negative number, minus 50 is a smaller potential compared to minus 100. To minus 100 is further away from 0, which would be your neutral value. And among all the ions that are present in the cell and in the extracellular fluids, potassium is the most important, regulating the resting membrane potential. And if you already work in some kind of health profession, you know that measuring potassium levels is of utmost importance, and changes in either increasing or decreasing the potassium levels can have very serious consequences. Okay, so I'm going to kind of go step by step what is going on to generate and maintain the resting membrane potential. Remember, this is a continuous process, so this is not step 1 or step 2. This is always going on. And one of the main players here is this pump, the sodium-potassium pump we talked before. So as you remember, the sodium-potassium pump takes 3 sodiums and ejects it from the cell, out of the cell, and takes 2 potassium ions back in the cell. So this is a continuous process. And how much of this is happening, it's going to depend a lot of the ion balance inside and outside the cell. We talked about potassium, and what happens is that in the cell there exist potassium leakage channels. So the pump is bringing potassium inside the cell. So there is going to be a concentration gradient of more potassium inside than outside. And because there are open channels, leakage channels, the potassium slowly goes out of the cell. It's just using the facilitated diffusion following its concentration gradient. So again, potassium is leaving the cell continuously, and potassium is a positive ion. So if potassium is leaving the cell, and inside the cell we also have negative ions, and these are usually proteins that cannot leave. That means that the inside of the cell becomes more negative. Okay, so going back for a second, the pump is bringing in potassium. The cell will have more potassium inside than outside. There is a concentration gradient. Potassium is leaving through the leakage channel. Potassium is positive. Potassium is leaving the cell, so the inside of the cell becomes more negative. Now, we have been talking about concentration gradient, which is related to the amount of substance in one side or the other side of the cell. But there is also something called the electrochemical gradient. Electrochemical basically is related to charges. And as you know, opposite charges attract each other. So positive attracts negative and vice versa. So we were saying that the positive potassium is leaving the cell, and the inside of the cell becomes more negative, and the negative inside of the cell starts to attract back positive potassium. And this has nothing to do with the amount of potassium. This has only to do with the charges. So at some point there is a balance, and that is how resting membrane potential is established. So there is going to be a balance between the potassium that is leaving because of the pump bringing in potassium and establishing a concentration gradient, and the attraction, the electrical attraction of positive potassium moving back because of the inside of the cell that is negative. And again, this is what we call a dynamic equilibrium. So there will be movement of potassium in, movement of potassium out, and this whole thing is balanced by the pump, and the rate of the active transport will be very much dependent of how much sodium goes in the cell. And usually in the cells, we are going to see that this resting membrane potential can change when cells are not resting. So there will be spikes of movement in and out of the cell, and then the pump will have a very important role reestablishing the resting membrane potential. But summarizing, it is a negative value. This inside of the cell is negative, and potassium will be coming in because of the pump, leaving because of a concentration gradient, and being attracted back because of its charge. And here is just a nice diagram showing you again all the players. So on the left, actually on both sides, you see this potassium leakage channels, in which potassium is leaving because of being more of it inside the cell, but on the other hand being attracted back because of the negativity of the inside of the cell. Okay, we are moving forward with this. We are going to see membrane potential again and again once we get into muscle and nervous system. But let's look at other functions regarding the cell membrane. So we talked about the glycocalyx before, and we talked about glycoproteins, which are part of these interactions between the cell and the environment. And cell adhesion molecules, as the name indicate, they have to do with adherence to either cells or to the matrix, extracellular matrix of the cell. And then we have membrane receptors, and these receptors can be receptors of hormones, of neurotransmitters, of growth factor, and they play an important role in cell communication. So expanding a little bit more of membrane receptors, and keep in mind this is just general introduction now, and then later down the road we are going to see specific examples of how these work. So contact signaling simply means that cells can touch each other or recognize each other, and this is very important in normal development. How, like when embryos develop, there is going to be a lot of changes, how parts of the embryo will become certain organs and other parts become completely different organs, and a lot of this has to do with direct cell-cell signaling. And another important part is immunity. So in immunity, the immune system is able to detect if cells are self or non-self, and then it will be attacking the self or non-self. Then we have chemical signaling, and here we have neurotransmitter, hormones, and paracrines. And basically what means that there are chemical messengers, and they are moving, for instance, inside the blood. In the blood, they go to a cell, they bind to the receptor, and then things happen. And we talked about insulin before. We can talk about neurotransmitters. We can talk about even drugs. Drugs are going to attach to normal receptors, and they are going to change how cells respond. And, a lot of medications, a lot of drugs are going to act at the receptor level. And finally, this is something that will come back again when we talk mainly in the nervous system, there are receptors called the G protein linked. Here the effect is a little bit slower, or let's say it's a little bit more complicated. What happens, the substance called the ligand is going to bind to a receptor, and then a so-called G protein becomes activated, and then things happen in the cell. Upon G protein activation, cells can grow, they can produce certain things, they can do different things. Again, this will make much more sense when we see specific examples. But again, these are examples of communication of the cell with the outer environment. And what you see here is how G proteins work, and I'm not going much into the detail. As I was saying, this is a slower process because it requires not only that the ligand binds to the receptor on the membrane, but also there will be a cascade of secondary messengers inside the cell. Okay, so with this we are moving on to the second general part of the cell, which is the cytoplasm. Again, all this was related to the membrane, the cell membrane. So the cytoplasm is basically the filling material of the cell. It is everything that is between the plasma membrane and the nucleus. And it consists in different things. One of them is the actual liquid material, which is often called the cytosol, which is water with different substances in them. Then we have the organelles, and then we have inclusions. And the difference between organelles and inclusions is that organelles have actually a structure and a membrane, and inclusions are mainly kind of storage places for the cell. Okay, so organelles can be membranous or non-membranous. The membranous are mitochondria, peroxisomes, lysosomes, ER, and Golgi, and non-membranous are the cytoskeleton, the centriole, and ribosomes. And I'm going to repeat some of the information I said in the first part. Now, we talked about the cell being this kind of factory, and we talked about the mitochondria being the powerhouse of the cells. And again, this is where most ATP is generated, and this happens via what's called cellular respiration, which is a process in which sugars, fats, and other substances are broken down to carbon dioxide and ATP. And what is really interesting about mitochondria is that if you look at the structure, they have this double membrane. They have an outer and an inner membrane, and they have this kind of structures called the cristae, and the cristae are like invagination of this inner membrane, and then there is inside them there is a substance called the matrix of the mitochondria. And what is really interesting is that mitochondria, billions of years ago, they were bacteria. And they basically become what we call symbionts. We started to live with eukaryotic cells. And this was a very, you know, mutually beneficial relationship. The bacteria inside the big cell were kind of protected, and then the big cell was, you know, benefiting from the large amount of ATP produced by the mitochondria. And for that reason, mitochondria contained their own DNA and RNA. We talked about ribosomes being the cytoprotein synthesis. We mentioned that they can be either associated to the rough ER or they can be free. And whether they are free or membrane bound, they are going to produce different kind of proteins. And we talked about the ER being continuous with the nuclear membrane, and there are two varieties, the rough and the smooth endoplasmic reticulum. And the rough ER, obviously, it's rough because it presents the ribosome, so this is the place where proteins, secreted proteins are produced, and some other proteins are also produced here, and phospholipids. Now, I mentioned that the smooth ER has different functions depending on what kind of cells, in what kind of cells they are. So in the liver, they have the role of detoxification, but they also do many metabolic reactions, lipid to lipid, cholesterol, breaking down glycogen, and so on. The liver is our main metabolic organ, so there is a lot of chemical reactions happening there. Also, in intestinal cells, they do a variety of processes related to fats, and in the muscle cells, they store and release calcium ions. The continuation kind of the ER is going to be this packaging and transporting center of the cell, which is the Golgi apparatus. And the way it works is that the proteins are released from the ER with the proteins inside. They fuse with one side of the Golgi, which is called the cis Golgi, and then the proteins kind of move across the Golgi where things happen to them. So again, imagine a packaging facility. So they are modified, concentrated, packaged, and then they are released from the trans Golgi and sent to wherever they need to be sent in the cell. We talked about the lysosome, the little stomachs of the cell, which are going to have very strong digestive enzymes. They can digest either outside cells of structures, they can degrade non-functional organelles, they can release different things depending on where they are. For instance, in the muscle, they can break down glycogen. In the bone, bone cells, they can release, they can break down the mineral bone to release calcium, and basically they just again break down anything that is either dangerous or not useful anymore. And peroxisomes are the one that defend us from free radicals. Again, this is the one that has kind of antioxidant capacity, so they are going to detoxify harmful substances. Okay, the cytoskeleton is this different types of rods, and they are of three kinds: microtubuli, microfilaments, and intermediate filaments. And I'm going to start from the smallest to the largest, smallest being the diameter. So microfilaments, if you look at the picture, their diameter is only 7 nanometers, so these are the thinnest filaments, and they are made of a protein called actin, and we are going to see a lot of actin when we get to the muscle. So they do functions related to cell motility, endocytosis, exocytosis. Then we have intermediate filaments, which are a little bit thicker, and you see that they look like ropes. So you have these proteins that are kind of wound to each other. So these are extremely strong filaments. They are a little bit thicker, 10 nanometer diameter, and they are going to be, well, the toughest filaments. So for that, they are kind of form the real skeleton of the cells. They are going to attach to desmosomes. Remember, the desmosomes were these kind of rivets between cells. So with the intermediate filaments, the cells are kind of attached to each other, and then they resist pulling forces. And finally, the largest one in diameter are the microtubuli. And observe that they are actually hollow tubes. So you have tubulin, and then tubulin they look like a string of pearls, as you can see, and then they form this helix, and you have this rather large tube and a diameter of 25 nanometer. And they do a lot of things. They do, they are part of the processes that happen during cell division. So they are going to move chromosomes. They are going to determine the shape of the cells. And interestingly enough, microtubuli are more represented in cancer cells compared to normal cells. It's a little bit like the cancer cells are made more in a hurry, so they use more of the microtubuli in the structures, and many cancer drugs are going actually to attack microtubuli, and that in turn, if you immobilize the microtubuli, the cell dies. Then there is some special motor molecules, and they have to do with movement inside the cell or contraction of the whole cell, and we will see this much more in detail when we get to muscles. The centrosome is also made of microtubuli. Actually, microtubuli generate from the centrosomes, and they contain something called the centriole. And the centriole, as you can see, it's kind of very nice looking. So the cross section is star shaped, and it's a tube that is formed by many microtubuli. Then we have cellular extension. So these are structures that are outside the cell. And there are two kinds, cilia and flagella. And the cilium, that's one cilium, and the cilia is the plural, is kind of short extensions, and they are kind of whip-like, hairy little extensions. And they are very important for example, in the respiratory system. So when the air goes in your respiratory system, and particularly I'm thinking here the pharynx, larynx, and the trachea, and down in the bronchi. Of course, the air carries particles, dust, bacteria, and so on. And the cilia are continuously beating, and they have these cells produce also a mucus, so they trapped all these particles, this dust, and then they move it away from the lungs. So by the time the air reaches the lungs, it becomes very, very clean. And so cilia are many and short. Flagella tend to be a long structure. It's like a tail, basically, and it's usually only one flagellum, and the purpose of flagellum is movement. So in humans, there is only one cell that has a flagellum, and it's going to be the sperm cell. And another kind of cellular extension are the so-called microvilli, and this is slightly different from cilia. So cilia are a separate structure. However, the microvilli are the plasma membrane that kind of projects these little fingers outside, extending from the plasma membrane, and then what you achieve with this is a huge increase in the surface area. So this is microvilli are very typical of cells that absorb. One example would be the intestines, the other in certain tubuli of the kidneys. And you are probably aware that there are certain diseases, like what's the name? Celiac disease, for instance, which is characterized by allergies to certain proteins, and those patients tend to have often diarrhea. And what happens is that in these patients, the microvilli are damaged or destroyed, and because the surface area is so much smaller, it means that the absorption of the nutrients is affected. Okay, so let's move on to the nucleus. We talked about the nucleus before. This is the place of the blueprint for the factory. So this is where DNA is present. It contains the recipes to make almost all cellular proteins. And most cells are going to have one nucleus. That's what is called uninucleate. There are certain cells that have lost their nuclei, and for instance, red blood cells have no nucleus. Sorry for the typo. And there are certain cells, on the other hand, that have many nuclei. So skeletal muscle cells, cells called osteoclasts, which destroy the bone, and some liver cells actually present more than one nucleus. And a reminder how it looks like. Again, it has a nuclear envelope, which is kind of a membrane enclosing the nucleus, which presents also pores. It contains chromatin, and inside we can observe a structure called the nucleolus. And the nucleolus, or nucleoli in plural, is it can be observed as a more dark staining body, and it's involved in RNA synthesis. We were talking about chromatin. So the DNA in the nucleus is combined with a certain kind of protein called histones, and also a little bit of RNA. And I think it's easier if I show it here. So you remember the DNA is a double helix, and see how the DNA starts winding around these histone proteins, and that kind of bead like structure is called a nucleosomes. And then itself, this thread kind of starts wrapping even more and more tightly, and finally it becomes a very, very condensed structure. So if we open the DNA molecule, it's extremely long, and it requires this very tight binding to to basically fit in the nucleus. Now let me just go back for a second. What you see down there, the number 5, that X-like structure is called a chromosome, and chromosome and chromatin are the same. So basically the chromatin during cell division acquired this very condensed dark appearance, and then we talked about chromosome, but it's basically the same thing, a combination of DNA and proteins. Okay, let's talk about the life of the cell. The life of the cell from the moment it originates, and remember, cell will come from another cell, so a new cell is produced after a previous cell division, and from that moment to the time that the cell itself divides, it's called the cell cycle. And there are two main parts of the cell cycle. One, which is the longest part, is the interphase, which would be the actual normal life of the cell, and then we will have the cell division or mitotic phase. Okay, so again, until the cell division, that longest phase is called the interphase, and in this phase the nuclear material is chromatin, so it's not going to be condensed, and there are 4 subphases, and I prefer to explain this here in this diagram here. So let's start from the beginning where it says G1. So the first phase of the interphase, which is the whole green part, and you can see this is the longest phase of the cell cycle. The first phase is called G1, which is the growth phase. Then comes a second phase called the S phase, and this also includes growth, but this is where DNA is copied. So DNA synthesis occurs here. Remember that cell division means that one cell becomes two cells, and each cell needs its own DNA. So before the cell division starts, the cell needs to make a copy of its DNA. And that happens during the S phase. Then comes a short phase. It doesn't seem shorter, but actually this is the shortest phase of the interphase, called the G2 phase. And this is kind of the phase of the final preparation for cell division. And after that comes the mitotic phase, which is where cell division occurs. I wanted to bring your attention to the checkpoints. There are two checkpoints in the cell during G1 and at the end of G2, so between G2 and M. And these checkpoints are kind of checking the integrity of the cell. Is everything okay? You know, does the cell look okay? There are, you know, chemical ways of the cell kind of collecting information if everything is the way it should be. If a cell is damaged for some reason, that cell is not allowed to divide. It should not be allowed to divide, because that means that more cells that are damaged are produced. Of course, we know that there are diseases or conditions where damaged cells, abnormal cells reproduce, such as in cancers, and cancer cells have lost that control. So that many of these control mechanisms, the checkpoints, are not correct in the cancer cells. And in the previous slide, there is also a phase called the G0 phase. And what it is that there are certain cells that once they become completely mature, they don't divide anymore. And examples of that are the neurons or nerve cells. You may have heard that, you know, you have to take care of your brain, because once the brain cells die, they don't grow again. And that is kind of true. So nervous cells very, very rarely grow again. Once they reach maturity, they move into this G0 phase, which means they live and they do their things, but they don't divide anymore. So this is the aspect of a cell during the interphase, and what you see on the right side, of course, it's a diagram of how, you know, the DNA is present in the chromatin. You have the nucleus, you have the nucleolus, and there are two centrioles present. On the left side, what you see is a very pretty fluorescent microscope picture, and it's staining different parts of the cell with different colors. So the blue is the nucleus, so you can see that the chromatin filling the nucleus is kind of homogeneous, and then the centriole are labeled in kind of this greenish color. Actually, the microtubuli are labeled in the greenish colors, and the red is staining for the filament, so you get the feeling of what the shape of the cell is. Okay, we were saying that the S phase is where DNA is replicated, and I'm going to make this very light. You'll be seeing this more in detail later on, but basically the DNA is replicated, so there is a copy made of each strand. So remember, the DNA has two strands, the two strands separate, and then complementary strands are made, and this is called the semi-conservative replication. And you remember that DNA has four bases, A, T, G, and C, and there is a complementarity. So A can only bind to T and G can only bind to C, and that guarantees that the new strands are going to be the replicas of the original DNA. And the enzyme that plays the most important role here is called the DNA polymerase. So you see how in the left you have the DNA molecule. It has the two strands. Then there are enzymes that unwind the DNA. It's called the helicase enzyme. So you see how now the two strands are separated, and you have the DNA polymerase adding new bases to the old strand. So you're going to have the old strand that acts as a template, then the nucleotides are coming in, the bases are coming in, the DNA polymerase is adding it. There is a color code so you can check that indeed adenine is going to, A is going to pair with T and C with G. And this is how the DNA is copied. So by the time cell division happens, there are 2 copies of the DNA ready to go. Now, mitosis, or sorry, not mitosis, the mitotic phase, which is the cell division phase, occurs in many cells of the human body, but not all of them. So we already mentioned nerve cells, skeletal muscle cells, and cardiac muscle doesn't really divide anymore. Tissues that are characterized by very active cell division are those tissues that are prone to damage or wear and tear. So think about our skin. Obviously our skin is exposed to the outside, so there is always cells falling off, basically dislodging from the outer layer. So the skin needs to be replenished continuously with new cells. The intestines are another place where lots of cell division occurs, because although it's inside the cell, the intestinal layers are exposed to kind of an outside environment, inside out. So if you think about it, the digestive system is opened to the external environment, so there's a lot of wear and tear going on in the intestines too. Okay, so the mitotic phase has 2 parts. One is the actual mitosis, which is the division of the nucleus, and then there is a second part, very short, it's called cytokinesis, which is where the cytoplasm divides. And within mitosis, which is again the nuclear division, there is a lot of confusion. A lot of people talk about mitosis as cell division. It's actually the division of the nucleus. Has 4 phases: prophase, metaphase, anaphase, and telophase. Now, the way you know what's going on is that you remember prophase. If you remember prophase, you are going to know telophase, because telophase is the opposite to prophase. And meta and ana are easy to remember. Okay, so let's see what happens in early and late prophase. So look to the left, and you see that something is happening in the nucleus. Look at the nucleus and see how you see something, that blue that was so homogeneous before, now it starts looking a bit different. And then if you look to the right, you see these condensed threads in the nucleus. And that is when chromatin becomes chromosomes. So chromatin is going to condense in distinct bodies called chromosomes. Then the nucleolus disappears. You don't see the nucleolus anymore. And then by the time the late prophase happens, there is no nucleus anymore, because the nuclear membrane disappears also. You see also how the centrioli, which were very together in the beginning, now they start separating. And so you see how they are moving towards the ends of the cell. And it's kind of hard to see in the picture, but there will be threads in between the 2 of them, and this is what is called the mitotic spindle. And this is the threads on which the chromosomes are being dragged along the cell. Metaphase is a little bit like the runners are you know, at the line of the starting line, lining up. So the metaphase is a very obvious phase, because all the chromosomes are going to be aligned in the exact middle of the cell, and this is called the equator of the cells. And then you have the centrioli on each end of the cell, and then you can observe now really clearly the mitotic spindle. And in anaphase, the chromosomes start moving. So what happens is, actually, remember that the DNA was duplicated, correct? In the S phase, the DNA was replicated, so you have a copy of the DNA. So in the anaphase, the chromosomes are going to split, and each copy of the DNA contained in the chromosome is going to separate and is being pulled by motor proteins towards the poles of the cell towards the centriole. And finally, in the telophase, again, it's the opposite to prophase. So now the chromosomes will unwind and become chromatin. There will be new nuclear membranes form. The nucleoli reappear. So now we are going to have two nuclei on each side of the cell. And then finally, the cytokinesis occur. And in animal cells, human cells, the way it happens is like it's almost like you're tightening a belt in the middle of the cell. There is something called the contractile ring. It becomes thinner or tighter and tighter and tighter until the two cells separate. Okay, as I was saying, the cell division has controls. So sometimes cells need to divide, and there are signals that are positive for cell division that we call go signals. Let's say that the cell has become too big and needs to divide into smaller cells. Or there are also lots of chemicals that are stimulating for cell division, and this happens a lot when, for instance, during growth. So when a child is growing, for instance, there are many hormones and growth factors acting on particularly muscle and bones and other cells in order to divide the cells, so it becomes bigger. Stop signals can include something called the contact inhibition, which means that cells don't really like to be piled up on each other, and if they start touching too much, they stop growing, and cancer cells has lost this inhibition. And there are also growth inhibiting factors that are made by genes that will repress growth. Okay, so what we saw before happens during the life of the cell, and particularly cell division. Now, we talked about DNA replication, and we talked about how DNA replication is required in order to make a copy of the DNA. Now, DNA is involved in other reactions and processes, and one essential process for a cell to survive is the ability to make protein. So protein synthesis is a process that brings information that is stored in DNA to protein. So remember, DNA has the instructions for protein synthesis. Now, how does this happen? In the DNA, we can find sequences called genes. So a gene is a piece of DNA, is a segment of a DNA that carries the instruction for one protein. Now, how does this happen? The DNA has a code of 4 bases. You have A, T, G, and C. Proteins can be made out of 20 amino acids. So that means that you have to have some kind of code to make the transition between 4 and 20. And the genetic code is based on 3. So 3 bases are going to code for 1 amino acid. And the way it occurs is that you have the DNA, and there is a process called transcription. And in transcription, what happens is that the recipe of one protein containing one gene is copied to an RNA. So again, in transcription, only one gene is copied. And this particular RNA is called the messenger or mRNA. Then the mRNA goes out the nucleus, and then it goes to the ribosomes. Remember, ribosomes were the place of protein synthesis, and then using those instructions in the RNA, proteins are made. And an analogy for this is, let's say that you have this extremely valuable, big, huge cookbook that you inherited from your grandma, and it's a very valuable cookbook, so you have it in a special place in your home. and then once in a while you want to cook a meal or some kind of recipe out of the book. So let's say that you take a copy, or you copy one recipe or one piece of paper, and you take only that piece of paper to the kitchen. You don't want to carry the whole big cookbook to the kitchen. So in this analogy, the big cookbook would be your the DNA. The recipe that you copy is the recipe for one protein gene sequence, and the paper where you copied it is the messenger RNA. And then you move to the kitchen, which would be the ribosome, where you cook. So this is where translation takes place. Okay, we talked about DNA. RNA, which is ribonucleic acid, is another essential nucleic acid. There are three types of RNA: mRNA, messenger RNA, ribosomal, and transfer RNA. And ribosomal RNA is basically an RNA that makes up the ribosomes. And transfer RNA are transporting amino acids, and they carry amino acids for the process of translation. Okay, so as I was mentioning, transcription is the copying of one gene from DNA to RNA. There are lots of regulation involved in this process, and the factors regulating is called transcription factors. The enzyme that does this is called the RNA polymerase, and it works exactly the same as during replication, except that in RNA we have uracil instead of thymine. So A will pair with U instead of T, and G will pair with C. And this is the diagram in your book that shows how transcription takes place. And that big bubble that you see there is the enzyme. The RNA polymerase is a huge protein. Binds to the DNA, opens the DNA where the important gene is present. And you see in the magnified diagram here how the DNA has been separated, and then that little blue line is going to be the mRNA being built by adding nucleotides using the DNA as a template. Translation is another word for protein synthesis. And we were talking about the genetic code. So the genetic code is a list of combinations of 3 bases. And okay, I need to put this correctly. I'm sorry. It says that in mRNA triplets are called anticodons. It's actually called codons. Anticodons are in the tRNA. Okay, so this is what you see there is the triplets corresponding to the mRNA sequence, and this is codons. And you see that there are 3 of them, and the letters that say pro, ala, asp, and so on are abbreviations of amino acids. So these are the 20 amino acids and their corresponding triplets. And you see that there are some special codons, start codons or stop codons, that will signal when the protein starts and when the protein ends. I really I really recommend that besides whatever videos I have uploaded in the website, you also go to YouTube and look for animation. This is much easier to see if you see it in motion. So what happens is that mRNA again has left the nucleus, goes to the ribosomes, attaches to a smaller unit of the ribosomes, and then it kind of aligns to the beginning, which would be the start codon. Then the bigger unit attaches, so now the ribosome is ready to go. And then tRNAs will be bringing amino acids one by one to the ribosome, and that way the amino acids that they carry will be attaching one to each other, and this goes on and on and on until the stop codon is reached, and then this amino acid chain is released. So looking at the diagram again, up there you see the nucleus, transcription happen, the mRNA is moving, going in here, the red thing is the ribosome. You see that it has two parts. The lower part is a smaller unit, the upper part is a larger unit, and you can see how the triplets or the codons are aligned inside the ribosomes. And let's say that you have, let's look at the middle one, you have CCG. So the complementary signal or complementary sequence for CCG is GGC, and that signal is going to be on the tRNAs, and the tRNAs are those little guitar-shaped molecules, and they are going to carry the corresponding amino acids. So you see that each of them is attached to one specific amino acid. So the amino acid, the tRNA comes in and attaches to the side, and then the amino acid coming in will attach to the chain that it exists. So again, this makes much more sense if you see it in action. But what happens is that it's always the last tRNA that has this long amino acid chain bound to it, and then finally when it reaches a stop codon, then the amino acid chain, the polypeptide chain, the protein is released from the tRNA and then leaves the ribosome. All right, almost there. So eukaryotic DNA has the DNA that codes for proteins, the DNA that has the genes, that is called the exon or coding DNA. Now there is a large part, actually this is the majority of the DNA, that is called the intron or non-coding or even junk DNA. And a lot of this actually codes for things. They can code for different special RNAs such as antisense and microRNAs. They have a regulatory role, or so-called ribose switches that are RNAs that can act almost like proteins as enzymes. And there are other really junk regions of the DNA. And you know, a little bit like you can think about it, you think about a computer hard disk that you haven't formatted in a long time, and you know how you start acquiring all this information, all programs, a lot of useless junk that kind of starts taking more and more space of your hard disk. And this is a little bit what's going on in the DNA with the junk DNA. And we just seem to have, human DNA just seem to have a huge amount of needless information. And so we're talking about the cell. But again, the cell exists in an environment, and this environment, these extracellular materials can be of different kinds. So we can have body fluids outside, and body fluids are actually very important in the sense that those are the few of the fluids we can actually monitor in a clinical setting. So if you think about blood plasma, we can figure out what's going in the plasma. We can even know what's going on in the cerebrospinal fluid. It's harder to see in the interstitial fluid, which is the fluid that is inside the cells. But body fluids are a very important way of cells to communicate and for humans to figure out what's going on in the body. Then we have a variety of cellular secretions that think about intestinal fluid, gastric fluid, saliva, mucus, etc. And then there is the cellular matrix. And this is very common, for instance, in connective tissue. Then you have harder matrices, such in the bone or cartilage. So these are basically filling material between cells.